the regrowing hair come from? Actually, Tournai is being based in Belgium. I guess all of you know where Belgium is, but Tournai is rather a small city, I would say across the road between London, Brussels, and Paris, somewhere in the center of Europe. Of course, Brussels is a big European center, and most of you have heard about the European administration. So if you look on the top right, the figure shows a man with a leg that has been amputated, but he's also an archer. He can hunt or perform sports. Now, the important point about this image is if you look here, you will all agree, I guess, that he is a handicapped person. He has a prosthetic leg that helps him in coping with the disorder. He's also performing very nicely from the physical point of view as being an archer. But this part of his body, the scalp, may or may not cause him some trouble in self-perception, but also in perception by others. And what we are about gathering today is about exhaustive measuring. Exhaustive means try to take into account all parameters intervening in the generation of alopecia. Now, these are strange images. They help us to remind about an object that we see on the background. These are two areas of a hairy man who painted himself about 100 years before computers came into the game. And he extremely nicely highlighted the influence of light. You see the left and right normally are receding frontal hairline, but they look very different in the global view. In the close-up view, we will see whether this is perfectly symmetrical or not. That's another issue. If we take a skin sample from the occipital area, we see that there are two terminal hairs here. There are three roots. One hair is not visible at the surface. And we see that they are implanted quite deeply in the dermis, reaching the hypodermis. This must probably mean something in terms of quality of the project, rate of growth, length, duration of the cycle, etc., producing what we call from the surface, which is level of view number 1, a thick terminal hair growth. If we look on the right, we see a quite rich hairy area, but most of the hair is miniaturized. There is only one thick terminal follicle that we see here. And we see that the depth of that follicle from the balding area is situated much higher into the dermis. So the depth is much less than the occipital scalp. We also see that the dermal hypodermal junction is modified and looks like a sclerotic tissue. We also notice, and scalp transplantation surgeons will notice that here we have a kind of expansion of the sample. If I take a punch, the fat cells will relax. And here it's exactly the contrary. We see a retraction of the specimen. So this probably all has a meaning, and we will try to see how we can measure what's happening in the depth by looking at the surface. So in summary, this slide tells us that thick terminal follicles seated deeply into the hypodermis dermal junction will produce a better hair during a longer time. Now, just maybe that's for the discussion. It's repeatedly asked during FDA discussions, but you count hairs, but what is the clinical relevance of your hair count? And the answer to that is just what is the threshold of what you can see? And nobody, I think, in the world today has really asked the question about the technology that has been used during FDA trials. We see hair on a photo, that's it. Now in 1994, so some years ago, we realize that we can look at scalp from different aspects. From the surface, that's our phototrichogram technique. Making a punch biopsy, you can section it vertically or horizontally, and you can extract information. Now, there is another approach, totally non-invasive or semi-invasive. If you isolate a little area on the scalp surface and you put crazy glue on it, you are able to extract all the hair roots at once. You see that most of these in the hairy side are seated quite deeply into the dermis. And when they finish their cycle, they will move up into the dermis to become telogen hair. They wait there for a couple of months, usually on average it's about 3 months, before shedding, that's entering the exogen phase. And in red, on this slide, you see that some follicles are less deeply seated into the dermis. And when they enter the telogen phase, the telogen club is situated higher up in the dermis. And this may signify something in terms of quality of hair replacement. If we move now to the right upper part, we have clearly a balding scalp. There is no many hair to see. And we extract all the hair and we see that the few hair that we can see is usually in telogen. It's very fine hair fibers. And the main question we ask in terms of treatment, is it really possible that we can revert this miniaturization process? Now, let's come now to what's published in this paper that has been published in Plastic Aesthetic Research. We see patterns. Obviously, this person would be rated as having no hair loss. And then we can see a pattern like in females or like in males. These are just translations in Dominique's view of the Ludwig pattern published in the 70s and Hamilton pattern published in the 50s. We will see what a pattern means in a few seconds. Now, in order to perceive, eventually to measure it in a right way, we divided the scalp in a number of areas. The 9 first areas are on the top of the head and the 9 other areas are on the periphery. We also comb the hairs in the midline, and we have the parted midline with numbers 1, 2, 3, 4, going from the posterior to the anterior part, and we do it in males and females. For those who have seen the video, I call this the Belgian wafer technology, and I hope everyone has had the chance to taste the flavor and appreciate the Belgian wafers. So creating an area with little squares reminded me about my youth when I ate these excellent Belgian wafers. So let's come back to the science. I do not know of many papers where at the same time someone investigated the coverage measurement and at the same time had a phototrichogram technique in order to see what's happening in the depth. And finally, a third original aspect is that we have a precise location of these phototrichogram sites on the global view. Finally, in terms of global perception, we developed the technology for real-time examination in the clinic without any major computers, etc., or cameras. This is a handheld device. You can look through this kind of grid, have a repertoire of areas, a scale of density. This is the schematic representation after combing the hair in the midline. And we have measured comparatively what we can do in vivo and what is possible to do on the screen. It's perfectly correlated. So this is a series of males that we show. If you take the circle, because all these are taken exactly at the same scale, if you take the circle as a limit, I would say these 3 people have a big head and these 3 have a smaller head. It doesn't matter much because over time you will always go back to the same fields. There is no confusion because if hair is growing, you don't know exactly where the limits of the hair is. This is for females. of course, in both genders, we were able to relocate, this is the white circle, this is the white patch, to relocate exactly where we perform the phototrichogram so that we can make a correlation study between a global aspect, a global evaluation, and the close-up analytical approach of the hair cycle. Now, in terms of global perception, you remember the Belgian wafer approach. We see, for example, in the first panel here, that there is almost nothing very different from a maximum coverage. When the situation is getting worse, we observe mainly on the top of the head that there is a decreased coverage. And finally, when we enter into the most severe cases, with the 2 or 3 here, the density or the coverage is very low. But at the same time, we realize that the periphery begins to be invaded by the hair loss process. Now, combing the hair in the middle means this grayish area is less dark than the hair combed in spokes of a wheel. That's an important message. When you go to a volcano, you want to measure the temperature of what is happening. The best is to enter into the focus of activity. That's exactly what we do with midline parting. Again, in Ludwig II type hair loss, this grayish area is less gray than the top of the head, which is already less gray than the crown. And finally, in the most severe cases, we have exactly the same process of going to the most affected area and parting the hair helps us to quantify more precisely. Now, I did some reflection that takes us 1 or 2 minutes about what is making a scale. Everyone has used SCORAD for atopic dermatitis or PASI score for psoriasis. And what we do, we mix apples and pears. It's thickness of the skin, it is redness of the skin, it is scaling. We add all this together and make an average of a severity score. I disagree since the beginning with this type of scales. Now, what about a scale in trichology? You can make a binary scale. There is hair or there is no hair. And if you do this as an imagination exercise, you will end up with a pattern. That's exactly what Ludwig did, and that's exactly what Hamilton did and the others who developed scales. So they make a kind of black and white image. What I imagine then the next step, the other extreme, would be to have a high-tech scale, because we have about 100,000 hair on the scalp. I would say there must be something like a 100,000 intervals. Each hair has a meaning and adds to the other hair. Now to come back between black and white, which is a binary pattern, or the most sophisticated 100,000 intervals, I don't think that the clinicians will be happy by using 100,000 intervals. So they make clinical patterns, and nobody asked himself the question about the equality of intervals and whether during treatments we can monitor the severity scales. And there are papers that say we cannot do in a very reliable way. And we all know from treating patients that you do not jump from a Hamilton 4 suddenly into a Hamilton 3 or Hamilton 2. So we try to follow the line somewhere in between 1 and 100,000 with our Belgian wafer approach. And that's where we are, from a 100% coverage to very poor coverage, say 20% is the least that we have measured. Now, from this scale that we show here, 100% to 20%, we used an abacus, which is a translation of one measurement into another measurement. So we translate coverage into the amount of hair that can be produced by unit time. This amount will tell us what is the percentage of the area that is being covered per unit time. And so we can derive the time it takes to have a complete coverage of the area. In normal subjects, it's about average range between 15 and 20 days. So what is important in terms of understanding what the abacus means, we transform one dimension into a dynamic dimension related to time. Now, this is the result of our analysis in slightly less than 500 subjects that we observed. And what is interesting is in between these two extreme limits, say between 100 and 20%, we can align, and that was a big surprise for me, that males and females with pattern hair loss or no pattern hair loss, they all line up very kindly in a regression line. So the system, I would say, works. We also published in the Plastic and Aesthetic Research paper some results of clinical trials with real people. But let's imagine that we see a subject, we evaluate the scoring as 50% of the scalp hair covering the scalp skin, which translates into it would take on average about 80 days for this person to cover the hair, to cover the scalp with hair. Suppose that after a couple of months with the treatment, you estimate that this subject has 70% of scalp coverage instead of 50%. So we have a benefit of 20% in terms of coverage. Now, in terms of time to reach complete coverage, we gain much more than just 20%. We change from 80 to 50% more or less, which is a 30% benefit. So the abacus helps us to get closer to a real dimension that may be of interest for the clinician, may be of interest to the patient. The therapeutic response in this theoretical example is good, but a Hamilton 4 with the dispersion of data will probably still generate a type 4 pattern. A Ludwig II female will probably remain a Ludwig II female, but the hair coverage will be undoubtedly better. Now, I'll go very briefly about this because it's not in the paper, but it's a new ID. If we rank order the density, for example, like here, and we put that in correlation with the least affected, the medium affected, or the worst affected patients, you will see in most of the graphs that the worst cases are always on the bottom. This is the opinion of the clinician. This is the opinion according to density. All food and drug approved trials are based on density. And I guess somewhere that the limit of detection is above 60 micrometer in terms of thickness of the hair fiber. But we can also measure the growth project in the hair roots. And we see that sometimes some people jump from one category to another. Anagen percentage, which is a reflection of the duration of growth. Very surprisingly, you will see that the thicker hair in a Ludwig II patients are growing very nicely, but their number is decreased and the amount that is going to be produced is less. The growth rate is slower, but those subjects with a no pattern or eventually a Hamilton 2, Hamilton 1 subject is having a very fast hair growth rate as compared to the controls. And that's absolutely amazing. So we begin to break down what we perceive as clinician in a diversity of hair productivity. When we talk about hair productivity, we'll say, Dominique, your tool doesn't help anyway. I say no, because with the close-up view, and that's something interesting for trichoscopy, there is a criterion that is almost abnormal in all patients, even those who complain and do not have a pattern, or ones that I cannot classify as a pattern. They have a substantial increase of hair less than 20 micrometers, which I would set as a diagnostic criterion for the patient. And also those subjects quite nicely fit. If you look globally at the colors, the distribution of colors is almost the same as what a clinician would say in terms of coverage of area percent which translates into time to complete coverage, with the top value here is 22 days, and the worst cases between 70 and 120 days. So it's interesting that we as clinicians can find some measurements that fit with the global perception, but the analytical approach gives us much more information. Now let's move now to the last part of my presentation, which is where do they come from? So this is a typical phototrichogram series of images. This is immediately after clipping. This one is 2 days later. Usually we have between 47 and 49 hours. And this is the overlay of this image on top of that image on a hair-to-hair basis. The red lines always represent length of hair at baseline, and the variation of color has no meaning. It's just for the computer to understand what is being growing and non-growing. We get rid of trichostasis, which is a hypothesis, by putting some polymer on the skin surface. We let the polymer polymerize, and then we remove a majority of exogen hair, which is after telogen, when the hair is loosely attached and about ready to get out. Now this A3 image we will transform by using a color code. You must remember probably your courses of geography, where you saw cities with a big circle, for example. These areas are very good in producing uranium or copper, or having big industry. New York is a big circle with plenty of money. So here we have little circles which represent the diameter and the amount of growth of miniaturized hair. This blue dots means bigger circles reflecting the diameter together with the amount of hair for intermediate, the amount of growth by intermediate hair. And in green, we see in this baseline image, a few hair follicles capable of producing a significant amount of hair. Now, summing up these 3 dot mapping techniques into one, we have in gray the follicular units that we can number out from 1 to 96, I think, or 98. We overlay the amount of growth, so it comes back to a kind of geographical representation. You will be astonished to see how much variation there can be in just 1 week. This is the same subject, same area, 1 week later without treatment. So you can see popping up hair growing, but 1 month later, all this thick hair, or majority of this thick growing hair, will have disappeared. Remember the biopsy I showed you in the beginning? There is a thick terminal follicle, but it's not performing very well or very long, or not on a long time. Now with treatment, you see exactly the same image, and you see a lot of terminal hair, including very thick ones, the yellow ones, that pop up. This is a combination of finasteride and minoxidil during 3 months. So we raise the diameter 60, 70, and even a small number of 80 micrometer thick hair. But those hair derive from pre-existing follicles. They are not a reflection of transformation of miniaturized hair into terminal hair. And that's another key message from our study, and also from a review of many thousands of miniaturized hair measurements. We have no proof that miniaturization is reverted by drug treatments. So to come back to our first global image and close-up analysis of hair growth and hair loss, and to go back to the basics in clinic, if patients complain about hair loss, you have to listen to their complaint, look at the hair and touch the scalp. The people love it, and for the doctors, in your hands, you have already some piece of information. We use the clinical staging, and we bring about new evidence that a continuum of scalp coverage scoring consistently translates into amount of hair that can be produced per unit time, and also the reverse, the time needed for total coverage. Transformation, number of hair, or diameter of hair, or growth rate into one parameter, which is productivity, the amount of hair that you're able to produce per unit time, is, I think, something very promising because it's perfectly correlated with the clinical observation of coverage. Now, one of the points that we may discuss later on is how to organize transfer of this quite easy technology either into clinics worldwide or through computers with eventually artificial intelligence or so-called artificial intelligence based on our experience. Now, normally I should give up and thank you for your attention. Thank you for reading the paper.